So for lesson two, we're going to be focusing on the plasma membrane. So in lesson two, our focus question is why is the plasma membrane an important structure of the cell? So you've probably seen the plasma membrane before. Another name for the plasma membrane you may have seen is just cell membrane. And again, both of these mean the same exact thing. So if we're talking about the structure of the plasma membrane, it is made mostly of phospholipids. So the molecules in a plasma membrane are lipids. And if you remember back from module 6, those are just fats. So lipids are large molecules that are composed of two main parts, glycerol and fatty acid. So here is our normal triglyceride that we have in our cells. You can see here is our glycerol molecule, and we have our three fatty acid tails. And again, this is a triglyceride molecule. Now, in our phospholipid, you can tell that a phosphate group right here is added and replaces one of the fatty acids. So we have a phosphate head and two fatty acid tails when we look at the structure of our phospholipid. Again, just a little bit of a so the plasma membrane is composed of a phospholipid bilayer, and this word bi refers to two. And in this phospholipid bilayer, two layers of phospholipids are arranged tail to tail. So this means the heads point outwards towards the watery environment, and the tails are pointed towards the middle. Now remember, since these tails are lipids, that means they don't associate with water very well. This is why our membranes associate to the inside to protect them from those watery environments. So phospholipids arrange themselves in a way that allows the plasma membrane to exist in a watery environment. So you can see tails are located in the middle. Our fatty acid tails are located where the water cannot get to them. And our phosphate heads are associated in the watery environment. So again, the structure of our phospholipid makes it very well suited to survive in our aqueous environment. So each phospholipid has two main parts. It has a head and two tails. So the phosphate group that makes up the head and is polar is attracts or attacks water, okay? Or attracted to water, I guess I should say. So our phosphate group, which is the head, okay, is polar, and this one is attracted to water, okay? The two fatty acid tails are nonpolar and they are repelled by water. So when we draw this structure, okay, we have our phosphate head, which remember is what we refer to as hydrophilic, meaning that they are or will associate themselves with water. And we also have two fatty acid tails. So these fatty acid tails are hydrophilic, or excuse me, hydrophobic, meaning they are afraid or do not want to associate them with water. Now, when we draw our bilayer, remember our tails will always point towards the inside. So again, since it's a bilayer, this is composed of two layers of these phospholipids. Again, phosphate heads towards the outside, fatty acid tails towards the middle in order to protect them from that aqueous environment. Now, the phospholipids arrange in this way because the polar heads can be closest to the water, and the molecules and the nonpolar tails can be furthest away from the water molecules. So you can kind of see this in our this lipid bilayer right there. Heads pointed out, tails pointed in, and again, this is all to protect those those fatty acid tails from interacting with the water. So what this does is it creates a barrier that is polar at its surface and non-polar in the middle. Now, this means that the water-soluble substances will not easily move through the plasma membrane because they are stopped by this non-polar region in the middle. Now, again, fatty acid tails do not mix well with water. So the plasma membrane can separate the environment inside the cell from the environment outside the cell, meaning they will never interact with water. Now, we also have a couple other components of the plasma membrane. We have cholesterol, we have proteins, and we even have carbohydrates. Now, when those proteins are found on the outer surface of the cell membrane, proteins will actually use to transmit signals to the inside of the cell. So if the cell gets damaged or the cell disassociates from another cell close near, these send out signals and communicate with other cells in order to perform some sort of task. And again, these are proteins on the outside surface. Now, we also have proteins in the inner surface, which actually are used to anchor the plasma membrane to the cell. So, if you take a look at a cell, there is these structures called cytoskeletons that actually will run through the cell. Now, proteins on the inside of that plasma membrane are actually used to anchor those and keep the cell shape. Other proteins can actually span the entire membrane, which creates a tunnel that allows some of these nonpolar and large molecules to move inside of the cell. So these structures, these giant proteins, they're called transport proteins. In other words, they're proteins that are a tunnel to allow certain things to come in and out of the cell. We also have cholesterol molecules that are nonpolar, and those actually mix in with the fatty acid tails of the phospholipid and prevent them from sticking together because if those tails stick together, they won't work properly. They'll leave gaps, they'll leave areas for these substances to enter into the cell that may not want to be in there. Finally, we have carbohydrates, and these attach to proteins and stick out from the plasma membrane to identify the cell to the body. So, carbohydrates are used as an identification mechanism to say, yep, that's part of this body, we're not going to attack it. If a foreign substance comes in that these carbohydrates are not familiar with, the body will naturally attack them and eliminate them. So, as you can see here, you can see some of these different structures that we have. So, obviously, we have our transport protein right here. You can see it's almost like a tunnel that allows these molecules to move in. Here are our cholesterol, and our cholesterol again is kind of keeping these tails from sticking together. Remember, our cell membrane is a phospholipid bilayer, two layers of these phospholipids with polar heads and nonpolar tails. We also have these membrane proteins that are used for identification, as well as carbohydrate chains in order for that structure to work properly. So if you remember, the phospholipids that are in the bilayer create a C effect. And it's almost like waves where these phospholipids are not necessarily rigid, but they're almost like free flowing. And you can kind of compare the phospholipids to apples that are actually floating in a barrel of water. The C concept is the basis for the fluid mosaic model of the plasma membrane, which states that the membrane is a free floating structure that is not very rigid. So looking at this, and we are going to model the plasma membrane and label each of the parts. So let's start with our plasma membrane. And again, remember, this is mostly composed of phospholipids. So these phospholipids are arranged in a bilayer manner where the fatty acid tails are associated on the inside. Now, we also want to make sure we are drawing our proteins. And one of the big one is the transport protein, which is almost like our tunnel here. So again, let's label these. We have our phospholipid, which is the structure that our phospholipid bilayer is made of. We have our transport protein, which spans the entire length of the membrane. And so we can add a couple more phospholipids, and we can use this chance to draw our cholesterol molecules, which are used to kind of prevent the phospholipids from sticking. Now, we also have other proteins which are associated on the outside or maybe even the inside of the cell, which are used, the inside one, to anchor the cell and provide structural support. And the ones on the outside are used in a signaling mechanism. And a lot of these ones that are used for signaling will actually have a carbohydrate associated on the outside of it in order for that identification process. So again, three main parts: phospholipid, proteins, cholesterol molecules that are used and are making up our entire plasma membrane. Now, the plasma membrane is really important with doing one thing, and that is to maintain homeostasis in our body. So, the process of maintaining balance in an organism's internal environment is called homeostasis. And homeostasis is essential to the survival of the cell, because if the cell was no longer semi-permeable and or things weren't able to move through the cell membrane, waste would build up in the cell. There'd be an inappropriate balance of molecules in the cell, and it would cause the cell to be very traumatic event and also potentially die. So the plasma membrane is a thin, flexible boundary between a cell and its environment that allows nutrients into the cell and allows waste and other products to leave the cell. The plasma membrane is so important in maintaining the balance that supports life for the cells in our body. Now, a key property of the plasma membrane is it is selectively permeable, which means that the membrane allows substances to pass through while others keep it out. So think of the cell membrane like a net, right? So fish are not able to pass through the net, but water can flow in and out. So water is able to flow in and out of the cell very naturally, whereas oxygen and glucose needs these transport molecules in order to get them in. Waste is able to leave, carbon dioxide is able to leave, but these are not able to enter. So again, selectively permeable, some things are able to come in, other things are not. So let's think about what would happen if the cell membrane were not selectively permeable. And let's use this to support your answer. So if the cell was not selectively permeable, the cell might be destroyed because waste could not leave and an inappropriate amount of a molecule might enter the cell. This is going to disrupt homeostasis dramatically and potentially cause the cell to die. So, in summary, the plasma membrane is made of two layers of a phospholipid molecule, which remember is a head with two tails, a phosphate head, two fatty acid tails. And they are arranged in a way that the hydrophobic tails will never be associated with water. Cholesterol and transport proteins aid in the function of the plasma membrane. Remember, cholesterol is used to prevent those tails from sticking together, which allows them to perform the semi-permeable function that they do. And transport proteins allow molecules that can't pass through the plasma membrane to enter to leave in an appropriate amount. And remember, our plasma membrane is a fluid mosaic model, which means it's almost like an ocean where these molecules are not set in stone, but moving freely, almost like the waves of an ocean. And our plasma membrane is selectively permeable. And it is selectively permeable as a property of the plasma membrane that allows it to control what enters and leaves the cell, which are really important in maintaining homeostasis. So hopefully this video helps you out, especially with lesson 2 in the plasma membrane. And again, this is Mr. O'Brien going over module 7, lesson 2.